been with U of T, but he moved to, you heard 80% now. He works for Google Brain. He leads that team. So I'll introduce David a little bit. Now we come back. This is all internal people now. Just now, our speaker was Professor Ross Taylor from Johns Hopkins University. So our second speaker for today is Professor David Fleet. It's we're fortunate having him because he's very hard to get. I actually had to go through Sheila to get an appointment with David because he is truly very busy. So David joined U of T actually in the same year as I did, 2004. But he is a lot more experienced because he did a lot before he came to U of T. He was with Xerox PARC in California. That was the 1999 to 2003. And then he was a professor at Queen's University. So he also served as department chair at UT Scarborough for 5 years. So a lot of us know the journal, the top line premium journal, PAMI. David was on the board for a long, long time. He's still on the advisory board of the journal. CVPR, ICCV, all these conferences, et cetera. I think a lot of our trainees join or know of. So David has been closely with those conferences as program chairs, et cetera. So he is a computer vision guru. I know David's name before he came back to U of T when I was a grad student, because he find papers, computer vision and things. And so I'm very glad to have David to speak to us today. And I'll give the floor to David. And afterwards, he'll have some sort of time for our trainees to pose a few questions. So without further ado, David, you can take it over.
Okay, so should I just share my screen?
Yeah.
Let me share, I'll just share my whole desktop. Can everyone see that?
Yeah.
How about that?
Good.
Okay, so I wasn't actually sure what to talk about today, so as is common in research, what we all want to talk about is the thing we're most excited about, and so what I decided to do was talk about some recent work that we've done, which I think is pretty cool. So you all have heard of the coronavirus. In fact, the circumstances in which we find ourselves today are largely determined by the coronavirus. And the key to understanding the coronavirus is understanding the structure of the spike protein on the coronavirus, which actually binds to an ACE2 protein in the human body, which allows the transfer of the viral RNA into the human cells. And understanding that spike protein is key to the development of certain vaccines and antivirals, like Regeneron, which you might have heard of, was developed based on understanding the structure of the spike protein. And there was another antiviral that was considered remdesivir, which was also based largely on our understanding, not of the spike protein, but the internal workings of RDRP, which is sort of inside the SARS-CoV-2 virus. And the key to understanding the structure is actually a computer vision problem, which is they take a bunch of images from these proteins and then they understand their 3D structure from running computation or computer vision algorithms on these images. And the techniques that were used to solve the spike protein were the algorithms and the software that we developed here in Toronto. And so it was kind of an exciting development, and I'm going to talk about that. But to get there, I actually want to go back 30 or 40 years and talk about some basic principles in computer vision and how we understand aspects of the 3D world from 2D images. So I hope this is more or less appropriate, but I want to just go back and start with some very basic elements. So this is a video that I first saw. A friend of mine showed it to me about 8 years ago. And it shows software that was being developed for autonomous driving back in 2012, 2013 by a company called Mobileye. It shows sort of what you would expect a vision system to do. It recognizes the other vehicles, it recognizes the pedestrians, it recognizes cyclists, and it understands the structure of the road, and it actually looks pretty straightforward, and does pretty much what you sort of intuitively feel vision does, which is, you know, you walk around the world and you simply see things and you see where they are, and it seems like a fairly effortless process, but it's not as effortless as you might think. In fact, going back, you know, 2,000 years, Aristotle is often quoted as saying vision is simply knowing what is where by seeing. In other words, we simply see stuff in the world. But in fact, vision is a lot more complicated than that. And one of the problems is that ambiguity is sort of intrinsic in visual processing, that any given image is often consistent with many, many scenes in the world. And one of the great tasks of computer vision is essentially to untangle that ambiguity and take what is a sort of sparse amount of two-dimensional information in a single image and infer the structure of a 3D environment. Let me give you another example. This is a video that a couple of buddies of mine took. This is Rob Fergus, who's at NYU, and it shows Rob. You can see him in front of a computer. He's got his headset on. He's working with his keyboard, etc. But this is a blurry image, and because it's blurry, you don't get to see all the details. Let me show you. Some of you may have seen this. Let me show you the full resolution version. So he's not looking at a computer monitor. He's looking at a waste paper basket. And the thing he was talking to was not a phone. That was his shoe. And his computer mouse, that's a stapler. And what he's got around his head, of course, are not headphones. But so the point is that the way in which we see the world relies tremendously on having models of the world and having all sorts of prior information which you bring to bear to disambiguate the intrinsic sort of ambiguity that exists within an image. Okay. And in fact, this was captured. This is not something new. This was captured. There's a well-known American psychologist, William James, and he has this quote that says, well, part of what we perceive comes through our senses. In other words, through your eyes, from the object. He says another part, and he said it might be the larger part, always comes out of our own mind. In other words, we're not just seeing what's where, we're actually making it up. That a lot of it is the form of inference. And there's a beautiful example of this from Ted Adelson's lab, Ted Adelson at MIT. And he came up with the following illusion, which is nice. It's often known as the checkerboard illusion. And it shows a very simple scene. And the idea is to look at these two patches on the checkerboard. And you can ask the question, what color are those patches? And by that, what I mean is how much light is actually emitted from those patches. If you were to take a photometer and measure the number of photons that are emitted toward your eye from those two regions on the screen, would they be the same or different? And of course, it's an illusion. And so although they look different, the answer is they're actually the same. And let me just illustrate that. I'm just going to slide this thing across. And those are the two patches if you occlude everything else that's going on in the scene. Okay, so what's going on here? How is it that those two patches are actually identical, but we perceive them as being very different? And to really get at that, you have to go back and understand the geometry and the photometry of how images are formed in the first place. So to start with the image formation process, you can think about a, here, let me do this. You can think about a surface in the world, and you think about some geometry of that surface, and that surface is illuminated essentially, the surface is illuminated by a light source. and once you illuminate the light comes from that light source and interacts with the material properties on the surface. And then some of the light from that surface then is reflected, and that goes into your eye. And this is a fairly complex process, of course, involving the material properties, the geometry of the surface, the form of the illuminant, the color of the illuminant, etc. But a very simple model for this process is called Lambertian reflectance, or Lambert's model. And that says that L, which is the light that you receive in your eye, can be thought of as a product of two terms. The first term, R, is the surface reflectance, or what's called the albedo. And that's the fraction of light that gets reflected to your eye from the light that actually is incident on the surface. So there's a certain amount of incident light, and the amount of incident light itself depends on the orientation of the local surface, and it depends on the direction of the light source. So it depends on the angle between N and S, and it depends on the intensity of the light source. The more the intensity of the light, the more light is reflected to the eye. And the lighter the surface, or the higher the albedo, the greater the fraction of light that is incident on the surface actually gets reflected back to your eye. And so with that simple model, we can understand Adelson's illusion, which is that the light that you see, the light that gets projected to your eye, depends on this product of the albedo and the incident light. Now, what you can see here in the image is that, and this is what your brain perceives, that part of this image is actually in shadow. So part of this image, your brain thinks, is receiving less light in the first place because it's being blocked by this cylindrical object from the light source. And in other words, so if you think about this patch here in the center, its amount of incident light may be lower. Let's say it's half of the incident light from this patch over here, which received direct light source. And so if H is actually lower by a half, in order for L to be constant, and I argued earlier that the amount of light that hits your eye on these two patches are exactly the same. If H here in the center is a half of what it is over here, then R, the albedo, must be twice as large in order to conserve the amount of light that hits your eye. And when R is larger, that means a greater fraction of light that's incident on the surface gets reflected to your eye, and that in turn means you perceive it as being a brighter surface. Now, there are many other sources. So effectively, let me state vision another way. Vision is a process by where you measure something like L, and based on that, you have to factor that measurement into the multiple causes of the light. And some of those causes relate to the amount of incident light, the geometry of the surface, the reflectivity of the surface, or the albedo of the surface, etc. So you're receiving one number, and from that you have to decide what product of numbers is actually responsible for the number you perceived. Now, there are many other sources of ambiguity. One of the big ones is the fact that when you perceive the world, or when you sense the world, you're sensing a two-dimensional projection of three-dimensional objects in the world. So this point on the tip of the flame gets projected through the pinhole of a camera. Normally we use a lens here, but in the ideal world, it's a simple aperture, and that gets projected to a point in the world or on the imaging surface. And what this projection means, that the three-dimensional world gets projected down onto the two-dimensional world, which means that information about the size and shape of objects is gone. It's distorted or confounded in some way. And part of the process of vision is reconstructing or inferring the three-dimensional shape and size of the world from these two-dimensional images. Now, one way that you can resolve, this is a great example of the size, shape ambiguity in images. This is the Ames room experiment. If you've been to the Ontario Science Centre, you will have seen this. The two girls in this image are twins. They're exactly the same. They're the same height. They're the same shape. They're dressed the same. I mean, there are slight differences. They're wearing different shoes. One has dark shoes, one has light shoes. But these are twin sisters. And I'm telling you, they're exactly the same size, but you don't perceive them that way. You perceive one as being small and one as being much larger. And the reason is because the shape of the room is actually like this. This girl over here on the left is actually much further away from the camera than the girl on the right. But this is a very strangely shaped room, interestingly, and you view the room from down here in the bottom. You view it from this peephole. So even though this is the shape of the room, you perceive it to be shaped like this. And that percept in and of itself means that you're perceiving the girl on the left to be much closer than she really is. And the only way to make sense of that is to assume that she's much smaller than she really is, because again, the size to which an image projects, the size to which an object projects onto your retina decreases with distance. So she actually is further away, so she projects to be much smaller, but you perceive the room such that she's the same distance from her sister, and therefore she must be perceived as smaller. And this is a bizarre illusion, but you prefer to see rectangular rooms, rectilinear rooms, rather than preserving the information you know to be true, which is that these two girls are exactly the same size. Okay, now there are all sorts of ways that we resolve ambiguity. And we resolve ambiguity. One of them is to have multiple views of the world. So imagine that you see a point of light in your left eye. You know that that point of light comes from somewhere along that line. That's what geometric optics tells us. Geometric optics tells us that if a point of light, a point in the world emits light, that light travels along a straight line through the nodal point of the lens and onto the imaging surface. Now, given that point in the left eye, we have no idea where it came from along this line. But if we can see another point in the right eye, now we can triangulate to find where that point is in the world. So as long as you only see one point of light in each eye, it's easy. What becomes more difficult is when you have a whole bunch of points of light and you don't know which point of light in the left eye corresponds to which points of light in the right eye. And that's where you have complex problems associated with stereopsis and the perception of three-dimensional depth. And to get into how the brain solves this, or how we solve this in computer vision, I want to break down the geometry and go into a little bit more detail. So here's sort of what the geometry looks like in the world. I have two images, the left image and the right image. I have nodal points of my camera. I have the left nodal point over here. Can you see my cursor? I hope so. So I have the left nodal point and the right nodal point, and I have one point of light in the world. And this point of light in the world projects through the nodal point of the left camera, and it appears in the image at the intersection of this ray here with the left image. Okay, and that same point also projects through the right image and appears at the intersection of that ray and the right image. And those two rays, they define a plane. And that plane includes the point in the world and the two nodal points. It also includes the two image points, and that's called an epipolar plane. And that's a sort of critical geometric object for understanding how the structure of two images relate to the three-dimensional world and to one another. Let me explain. So go back to this original ray of light that gets emitted from XP, my point in the world, onto the left image. Any point along that line will project to exactly the same point in the left image, but they project to different points in the right image. But you can prove that any point along that ray from the left nodal point to my point XP in the world, any point along that ray projects into the right image such that the intersection of those projections with the right image defines a line. And I know that to be true for the following reason, that any point along this ray that goes to the left nodal point, any point along that ray, if I project it through the right nodal point, it lies in the epipolar plane. And the intersection of the epipolar plane with the right image plane, the intersection of any two planes is a line. And so that tells me that any point along this line to the left image must lie somewhere along a line in the right image, and that's called an epipolar line. And it's determined solely by the geometry of your two cameras, in other words, where the cameras are and the directions they're looking, and the location of the point in the left eye. So if I know the location of this point in the left eye, this tells me that the corresponding point in the right eye isn't anywhere in the image, it's only along a single line. And that's important because what it tells us is if you want to find the correspondence between a point on the left image and a point on the right image, I don't have to search anywhere in the right image, I only have to search along a line. But it's a very special line. It's an epipolar line, and every point in the left image has a distinct epipolar line that it needs to search. Okay, but understanding that geometry greatly reduces the ambiguity of figuring out what point in the right image corresponds to what point in the left image. Now, if I take a different point in the world, let's call it XQ, I get this XQ, that projects to a different point in the left image and a point in the right image. And given this point in the left image, I now get a new epipolar line. And it turns out that what you can prove, it's not hard to prove this, is that for any point in the left image, there exists an epipolar line in the right image, and all those epipolar lines converge at a single point called the right epipole. And similarly, symmetrically, for any point in the left image, there exists an epipolar, sorry, for any point in the right image, there exists an epipolar line in the left image, and all epipolar lines in the left image converge on a point, which is called the left epipole, and the right and the left epipoles live on the line. It could be at infinity, but they live on a line that goes through the two nodal points of the camera. So in total, what this tells you is almost everything you need to know to solve two problems, which is one, how do I know where these two cameras are in the world? How do I know? And the answer is, once you know enough corresponding points in the two views, those corresponding points, along with the geometric constraints that I've outlined, are enough to actually uniquely infer where the cameras are in the world. Because once you know where the cameras are, you will have you have these constraints, which are these epipolar constraints, that all the correspondences between the left and right views must lie on epipolar lines, and all the epipolar lines must be convergent on the left and right epipoles. And so the totality of all these constraints, combined with the image constraints that tell us that matching points should have similar colors and similar textures, similar appearance, those allow us to basically calibrate cameras, and they allow us to infer the structure of the world. And there's a beautiful demo of this which came out about 15 years ago by Noah Stavely, Steve Seitz, and Rick Zeliski, who were working at Microsoft on the time, and they built an application called Phototourism. And so what they effectively did was just a minute. Let me find out where I am in my notes. Yeah, so what these guys did was they basically chose a place in the world, like the Tivoli fountain in Rome, in Italy. They searched up thousands and thousands of images of this one place. They identified sort of interesting, distinct fiducial points in these different views, and they figured out which images seem to share common points. And then between those images, they essentially use the epipolar geometry to figure out where the cameras were in the world and where the depth of all these points were in the world. And let me just show you a demo of what this allowed them to do. So this is Tivoli fountain. All these little prisms you see at the bottom, those are the inferred locations of the cameras. And in this case, I don't think they had that many cameras. I think they only had about 100,000 points and hundreds or maybe 1,000 cameras. But what that allowed them to do is for any given camera, it allows them to figure out where in the world it was from. And they built a three-dimensional model of the world. A year or so later, they took this to even larger scale. So this is something like 5,000 or 10,000 images of the city of Dubrovnik. And each of those little prisms is, again, one of the images they found. And what I'm showing you is basically their three-dimensional map of the environment in terms of a point cloud, which are all the corresponding points that they were able to find among these different views and rendered as a three-dimensional point cloud image. And this is one of the very, very early large-scale reconstructions of 3D environments that went hand in hand or actually preceded a lot of the early SLAM work, where you get dense reconstruction from 3D images. Now, more recently, what people have been really interested in is the semantic alignment problem, which is not just finding corresponding points in images of exactly the same scene, but actually trying to figure out semantic correspondence, or find me points in different images of different instances of objects from the same class that correspond in a semantic sense. So find me the points on the beak of the seagull that match the beak of the hawk, or find me points on the tail feather of the seagull and the tail feather of the hawk, or find me points on the car, on the wheels, or the windshield, or the rear view mirror, or the top of the car, and similarly with the bicycle on the bottom. So there's been a lot of work in the last 3 or 4 years on trying to figure out how to learn what kinds of points in images for a given class of object can be identified and aligned with other instances of the same class. And that goes a long way to being able to understand now not just the rigid structure of the same world seen in images, but to understand the semantic structure in 3 dimensions from a single image of a known class of object, where now what I'm doing is effectively I'm inferring some of the 3-dimensional structure of semantic features on an object now from a single image. Because I'm using the relationship between the points in that image and all the other objects in that class that I've seen before. And this has been taken even further in the last couple of years. There's a guy named Pete Florence who did a very nice PhD recently, where he took this idea of semantic correspondence and extended it to what you might call continuous semantic alignment, where what you actually do is interpolate these alignments. So at every point on an object, you know the alignment of that point on that object with other objects of the same class. And so what this is showing here are just some baseball caps. And what's shown on the right is a colored visualization of the correspondence, where parts of the hat which correspond are all in the same color. So the red part of the hat is the back of the hat, the blue part is sort of the middle here, etc. And you could do the same thing with other objects. So down here at the bottom are the same thing with shoes. And this is now important because now you know for any given shoe, what point on the shoe is the tongue of the shoe. And they were able to combine this with a robotic system and give the robot some basic instructions like pick the shoe up from the region of the tongue of the shoe in the middle. And you can see the robot sort of looks to the right and the left, again, because in order to get good three-dimensional information, it's good to have different views of the scene. It can then do the semantic correspondence and pick up the shoe near the tongue of the shoe. Okay, so that's in a very relatively short time. Sorry, I don't have my clock in front of me, so I don't know what time it is. But in a relatively short time, that's just going from images of the world to three-dimensional locations or fiducial points on objects. And this extends not just for rigid objects or near rigid objects, but people have done the same thing for articulated objects. Here they're doing it for human pose, where they're identifying these fiducial points, semantic alignment of joint locations on different people in three-dimensional scenes from video. And this has been further used actually by Sergey Levin's group at Berkeley for trying to learn control policies for robot motion. And the idea here is to track a motion of an object, something like a dog, in one view. And by being able to find these semantic correspondences, you can then go into the virtual domain and think about targeting the motion you see onto a reference motion of an object in simulation, for which you can then learn a policy, and then with some domain adaptation to close the gap from simulation to the real world, they were able to train policies on real world robots. Okay, so that's sort of what I wanted to get to in the introduction. That's like a very, very whirlwind tour of some of the work that's gone on over the last couple of decades in three-dimensional reconstruction of the world from two-dimensional images. And what I want to now do is go from large-scale reconstruction at the scale of cities down to the scale of biomolecules. So suppose we want to do the same thing with biomolecules. Suppose you want to understand the structure of a protein down at atomic resolution. And by atomic resolution, I mean down to the scale of angstroms. So 1 angstrom is 1/10 of a nanometer. So 10 to the minus 10th nanometers. And that's the scale at which you can discern a single water molecule or discern a carbon atom, for example. And so that is essentially the goal. And as I've talked about earlier, this is a cartoon of the coronavirus, and this on the right is the spike glycoprotein, or the S protein of the coronavirus, and that is key because that's the part of the coronavirus which is in these purple spikes, which actually attaches to proteins on human cells. So that is the site of attachment. And by finding antibodies that neutralize or that bind to the ends of the spike protein, you can effectively neutralize the virus. And that's what people did in, early this year in amazingly short amount of time with current technologies. Now, the typical way that people have solved for the structure of biomolecules down to atomic resolution in the past has either been NMR spectroscopy or X-ray crystallography. And the problem with NMR is it only works for super small biomolecules, molecules that are maybe 100 times smaller than the typical macromolecule, like this virus or a ribosome or proteasome that exist in the human body. X-ray crystallography does work with large particles, but it only works with particles that can be crystallized. So very regular molecular structures. And the problem there is it's sort of well known that only 4 or 5% at most of the proteins in the human body can be crystallized. So most of the proteins that are of interest for drug discovery, for example, cannot be crystallized, and so they're outside the scope of X-ray crystallography. And this is where a new technique called cryo-electron microscopy came into being. And the basis of cryo-EM, or electron cryomicroscopy, it sort of works like this. You first purify the protein in solution. Typically what you do is, and again, this is the biochemists do this, they are able to purify the protein in typically a water-based solution. And then what they do is they spread that solution on a carbon sheet so that it's extremely thin. Typically, it has to be around 100 nanometers or thinner. And then it gets frozen in a cryogen, typically liquid ethane. And once it's frozen in the cryogen, it freezes all the particles in place and keeps them in place so that you can put the sample into the vacuum of the electron microscope. Okay. Now the reason that it has to be so thin is sort of twofold. One is it greatly simplifies the imaging model, and two, it means in any given location in an image, you know that there's only one particle, because you can only sort of fit one particle in a thin film that's only 100 nanometers thick. Now, the one key thing to remember here is that when you image a bunch of biomolecules with an electron beam, you also destroy the biomolecules. So macromolecular structures, or biomolecules, proteins, are actually very sensitive. And when you inject a bunch of electrons, you change the energetic state of the particle, and therefore over time you degrade the electrochemical bonds that exist. And so typically when people take images in the electron microscope, they have to use very, very low doses of electrons. And what this means is the images are very noisy. Let me show you what the images look like. Oh yeah. So this is just a cartoon picture which tells you what's going on. So we have a layer of ice. Basically, it's vitreous ice. And then within that ice, at any given location, you have one particle, and the particles are all at random orientations. And you're going to image that with an electron beam onto a sensor. And this gives you a bunch of images. And this is one micrograph, which shows there are a few tens of images in this micrograph. And this has been low pass filtered and contrast enhanced so you can see them, but they're still hard to see. Here's what the images of the individual particles look like. So what I've done is I've gone into this image and cropped out a few individual particles, and they're sort of all you see is this really light silhouette of these particles. It's sort of dumbbell shaped. So it has two sort of darker circular blobs, and those are the particles. And what our goal is basically to take those particles, and they're extremely noisy, and to reconstruct the molecular structure of those, of the images of that structure. Okay, so that's the basic idea. Let me just let me take a quick break just for a second. Are there any questions that people have at this point? Sorry, I know I'm covering a lot of stuff.
It's wonderful. I'm just wondering how an accurate map can be built without knowing accurate output. That's what I'm thinking.
Yeah, it's a great question. Basically, the way it's going to work is like this, and I have a whole bunch of slides on this, but I wasn't going to show them. Basically, the way it's going to work here. Let me stop sharing for a second. Oh no, maybe that's dangerous. Don't play with technology. Basically, the way it's going to work is we're going to use a form of cross-validation. So what we're going to do is we're going to build a model with a subset of the data, and then we're going to test for the consistency of the remaining data. And we're going to do that in multiple ways, and that way you get internal measures of self-consistency. So it's a form of sort of almost k-fold cross-validation that we're going to be using. And that is the only tool that the biological community has available right now, is to use these statistical methods of cross-validation. So, I mean, the other tool is you show your work to a biologist and the biologist says, oh, that looks good. And that actually works extremely well.
So in the very end, David, how can one do the ultimate validation in the end?
Well, so what people have done is there are proteins that have been solved with X-ray crystallography. And so those provide benchmark tests so that when you solve it, in other words, that provides ground truth. And so that is one source of ground truth. Another source of ground truth is with the MNR or MNR on really small parts of the protein that were able to be decomposed and imaged with spectroscopy. And so there are ground truth sources of information, but beyond that, it's sort of cross-validation and your consistency. So typically we know the polypeptide sequence of these different, we know the sequence of amino acids. And so if the sequence of the amino acids fits this, what we're basically solving for is an electron density map, or the Coulomb potential of this three-dimensional field, right? We're effectively solving for the electron density as a function of three-dimensional positions. And if you can fit that really well to an actual sequence of amino acids, so if I give you a folded molecule, you know the attractive forces of every atom, and you can then figure out or simulate the electron density field or the Coulomb potential field. And so that gives you another way of comparing with, that gives you another source of ground truth, right? For any molecule that you know the fold for. But you're right, it's an interesting problem, and the biologists tend to be very skeptical. And so you have to do extensive amounts of sort of validation to show that these techniques are good. So what I want to do for the last sort of 30 minutes of the presentation is just show you how we formulated the problem and show you some of our results. Okay, so let me jump to that. Okay. So, before I get to that, actually, sorry. So, I mean, for any of you who are interested, there are all sorts of challenges in this space, and I'll just mention some of them. So the first one is these images are incredibly noisy. How do you even find the particles in the first place? When I first came across this problem, a PhD thesis would often, and this is 15 years ago, a PhD thesis in this field would mean a PhD student figures out how to biochemically purify a protein. Then they would collect a whole bunch of data, and then they would spend months, I mean, literally weeks and weeks of time going through these micrographs one by one with a mouse, picking out individual particles. Now, it turns out with deep learning, we can pick particles out of these micrographs much easier today. But again, these images are incredibly noisy, so just finding the particles is difficult. It turns out that when you image these particles, when you take an image of this frozen sheet, it turns out that there's actually motion. That with these CMOS sensors, you can record images at multiple times at about 30 hertz. And it turns out that the whole ice sheet deflects and moves a little bit when you image it. And so the next thing you have to do once you find the particles is do motion correction to stabilize them. And that allows you to average across the video for different frames, which reduces the amount of noise. And the images I showed you earlier are already averaged across multiple frames that are already noise reduced. Otherwise, within any single frame that you image, at most you get one electron count. So you're literally counting electrons in the detector itself. The next thing you have to do is take all these images and find an initial structure. And in the past, what people always did was used a lot of prior knowledge to specify an initial guess for the optimization. What we've been able to do is come up with a technique that finds an initial, what we call an ab initio structure, which is totally blind, requiring we actually start with random initial guesses and then run a form of stochastic optimization, which tends to empirically give us good starting points for then a more detailed high resolution refinement. Now, most of what I'm going to tell you today assumes that these particles are rigid, that what you have is hundreds of thousands. Often right now we deal with millions of images, and we assume that every particle in those images is identical to every other particle up to an unknown rigid coordinate transform. In other words, we know that we have the same particle, but we don't know the direction from which we're viewing it. In reality, that is actually a bad assumption, because it turns out, first of all, that these particles are very dynamic. They're always moving. And so what you get is a degree of conformational heterogeneity. So in other words, these particles are not actually rigid. They're actually quite flexible, and they're always moving. And how you accommodate that motion is something I'll talk about. You also get compositional variability, so that some of the particles will have a piece missing in some of the images, right? And you'll get other structure in, like, some regions of the particles will be quite rigid, and other regions of the particle will be quite non-rigid or unstructured. And so everything I'm going to show you initially assumes rigidity, that all these particles are identical, just in exactly the same way we looked at epipolar geometry. And then what I'm going to show you later are some results where we assume heterogeneous variability and compositional variability, where these particles vary from image to image. Okay, so that's basically what I'm going to show you over the next half hour. So the good news here is that we have a pretty good generative model. So what this image shows is that, assume we have a three-dimensional density, electron density V, and we know that what happens is, for any given image, this V is rotated and translated with respect to the coordinate frame on the microscope shown here on the right. And it turns out that with a sufficiently thin sheet of ice, the projection onto the two-dimensional image can be viewed as an integral projection. So all we're doing is integrating the density in the direction of the electron beam, and that gives us two-dimensional image, P, which is P for projection. And then that projection is essentially corrupted due to the point spread function of the microscope and due to noise. And what we're going to assume is Gaussian noise. We're going to assume that we've averaged enough individual frames of Poisson noise that we get a pretty good approximation to that a Gaussian noise gives us a pretty good approximation. So our images are a point spread function corrupted two-dimensional projection plus Gaussian noise. So what do we know and what do we don't know? We know the images. What we don't know is the volume, the density, and we don't know for every image the rotation and the translation of the particle in that image with respect to the camera coordinate frame. So assuming we have 1 million images, we might have something like 100,000 unknowns in the density function, and then we have about 5 million nuisance variables, which are the rotation and translation elements. And what we need to do is somehow get rid of those nuisance variables in order to estimate the volume V. And there are two ways to do that. One way is essentially what we call a marginalized likelihood. So what we're going to effectively do is define an objective function, which is solely a function of the unknown three-dimensional density. And what we're going to do is try and optimize that density so that it's as consistent as possible under a likelihood model. And our likelihood model comes from this generative model. So that generative model tells us a probability distribution over images conditioned on the volume. And the way we get that is by marginalizing out or taking expectation with respect to the unknown translation and rotation. So effectively what you have to do is find a probability distribution for each individual image. You get a probability distribution, a posterior distribution over the rotation and translation, and you're going to numerically integrate that out. So now if you had 1 million images, you have to integrate out 5 million parameters. So that's what's involved in this approach. The other approach is a maximum likelihood approach where you say, we're not going to integrate over this 5-dimensional space. Instead, we're going to optimize over that 5-dimensional space to find the rotation and the translation that are maximally consistent with each image given the volume. And by putting this maximization within our empirical loss here, we're able to optimize this V. But for both of these problems, you either have to integrate for every image over this 5-dimensional space, or you have to optimize over this 5-dimensional space. And one way or another, those are really hard problems. And one of the key results we came up with about 3 years ago, which made a big difference, was actually to derive a really efficient bound on the alignment error you get between an image and a particle. And that bound is going to be very useful for doing the optimization and for doing this integration here. And let me just tell you a little bit about the bound. It's actually not much, it's not hard at all. When I say alignment error here, all I mean is the negative log likelihood, because that's what we're really working with here. And the idea of the bound is that if you actually get rid of the high frequency structure and just look at the low resolution structure, that gives you already a pretty good idea of how this particle is oriented in this image. This particle, by the way, is an ATPase. It's an enzyme associated with energy production in living cells. And so it's a membrane protein. So this protein, you can already see that it's oriented like this. So that gives you a really good constraint on how the orientation is by ignoring the high resolution structure. The way we define the bound is we basically said, if you actually compute the negative log likelihood only using the low frequency components of the image, and all you do is bound the error in that based on the high frequency structure, then you get a very smooth and efficient, or at least cheap estimate of what the posterior distribution looks like over rotations and translations. Let me just show you pictorially what this looks like. This shows you for this image here, for this image up above on the left, and this ATPase below here on the left. This shows you the negative log likelihood, or if you want to think of it, the squared alignment error between this image and this particle as a function of as a function of position on the sphere. The sphere has just been flattened out, so you can see all the different functions. So as a function of the orientation of this particle, this shows you what the error looks like. And you can see, of course, the maximum, the lowest error is here in this little red dot. But if what we do is we only take a few low frequency components of the image and we calculate the error surface, and then from that, a bound, we get something that looks like this. So using only these low frequency components, we get this lower bound on the error. And what this tells us is there's no way it can be up here near the poles. we know that it's somewhere in this region or here or here or here. Okay, and this also, it gives us 2 bits of information. It tells us where we look if we're trying to do optimization, and if we're doing numerical integration, it tells us where to put the points that we're going to use for numerical integration to do it efficiently using Monte Carlo techniques. Okay, so so let me not go into a lot more detail. Let me show you some of the results now. So the way we do ab initio reconstruction is we marginalize over everything. In a true Bayesian fashion, the more you marginalize, the more you take all of the uncertainty in your model into account. And all we do is we form this marginal likelihood where we use Monte Carlo integration to get rid of the rotation and the translation. And then what we do is stochastic gradient descent on this objective. And there's a whole bunch of tricks under the hood to do this efficiently. And I'm not going to talk about all of them. And they're all really critical, but I just want to jump to the results. So here is a random initial guess of a smooth structure. And what I'm going to show you now is just a video, and I hope this comes through the Zoom channel. This just shows you the first few steps of initialization, getting down to a structure that is correct to around 12 or 15 angstroms. We can now do this in about 25 minutes. So prior to this work, this is a step that would have taken typically a few weeks of computation on hundreds of computer cores. And through the use of our efficient bound and the stochastic gradient descent with a bunch of tricks for optimization, we were able to get this down really efficiently. This is a large ribosome structure. It's actually a ribosome structure of a proteasome, which is it's a parasite which causes malaria, and they want to know the structure of the ribosome so that they can find antiprotozoan drugs which will disrupt the function of the ribosome and therefore stop the spread of malaria. But let me continue. This is an ion channel. It's actually a protein that it's a signaling protein that tells the inside of a cell something about the outside of the cell. And this one is part of the somatosensory system that tells the inside of the cell something about the ambient temperature of the cell. This is actually the protein that binds to the agent in chili peppers that gives you the sensation of heat when you eat spicy food. And again, here's the structure of this TRPV1 ion channel, getting down to around 12 to 15 angstroms, again in about 25 minutes. Once we get to this point, we get a pretty good initial structure. We then switch the optimization to do an iterative form of refinement. What we find is that once you get a pretty good initial guess, the posterior distribution over the rotation and translation become very well peaked. So we don't need to marginalize anymore. All we do instead is an optimization. So what we're going to do now is switch to the following result. We're going to be optimizing this function, which is a function which is the likelihood or the negative log likelihood of the image given the density model, which we're trying to find, and given the best possible orientation and translation between the current best guess about the 3D density and our images. And so what's going to be hard in this problem is solving this minimization problem. How do we find the best translation and rotation for a given image given our current guess as to the 3D structure? And the way it's going to work is the iterative refinement process is going to have 3 steps. The first step is to find the best rotation and translation. The second step is then to use a common tomographic reconstruction technique to get a noisy estimate of the density. And then the third step is essentially going to be to denoise it. And that gives us a new estimate. And now we go back to step 1. So we're going to be repeating steps 1 through 3. And I'm going to tell you mainly about step 1 here. I want to just tell you a little bit about that. Now, what we can use, once you have a bound on any function, if you want to optimize it, it's good to think about a branch and bound technique. Branch and bound is good because it's incredibly efficient, and provably it can be used to find a global minimum of your error surface. And here's how it works. So there's our error surface. It's extremely costly to compute, but we know it exists. But what we have is a lower bound on that error surface that's really easy to compute because it only involves a few Fourier coefficients. So we know it's fast to compute and it's super smooth. Once you have that lower bound, you know that the true error must be above that. So here's what we do. Because it's super smooth, we first find a point which is close to the minimum of that. And we can do that with exhaustive search because it's super smooth. Once you find that, you can calculate the true error, not everywhere, but only at that point. So now you know the following thing. You know that the true error cannot be lower than this red curve or this orange curve, but you also know that the best possible error is not going to be any larger than the error you've already computed. So you know that the upper bound on the error is this blue line, and the lower bound on the error is this orange line. So now you've reduced the feasible range of orientations to this small limit. And now what you're going to do is compute a slightly better bound, but only in this region, and reduce the feasible alignment. Let me show you what that looks like. So let's say this is the true error, and here's our initial bound. So we find the minimum, that's there, and then based on our branch and bound procedure, we calculate the true error only at that point, and then based on our branch and bound, upper bound and lower bound, we know that the true minimum is only in this colored area. We can immediately remove all this area that's white. Then we find the minimum for that, and we get a new bound, and that gives us a new feasible area. We calculate the true error at the minimum of that point, and that gives us a new area, etc. And this is what we end up with now. We do this whole iterative procedure. And this shows that this ion channel that I mentioned earlier. And also what I've done is I've aligned the true molecular structure, what I've called the docked molecular structure, onto the density map to show you effectively how accurate it is. Oh, in fact, here's another way. So this shows you the like a level set of the density map aligned with the true molecular structure. So for this structure, the true molecular structure was known ahead of time, and this shows the consistency of the molecular structure. This is another example. This shows the ab initio structure of the ribosome, and this is the actual ribosome that we compute. And this has hundreds of thousands of atoms in it, all of which are resolved to pretty good resolution. So we've been able to identify all the amino acid structures. Again, this shows, but it's not, I think this is probably hard to see, the just a single alpha helix from this ribosome. Okay, oh, here's one more example. This is a large proteasome, which is a protein in the body that's responsible for regulating the amounts of different proteins in the body, or picking up and decomposing misfolded proteins. And again, you know, we get great structure. We're down to around between 2 and 3 angstrom resolution, which is the resolution at which you can, what you really want to get, for example, in this alpha helix on the right, what you're really trying to find are these amino acid side chains. Those are the key properties of the biomolecule that allow you to look at properties like binding, other functional properties, and where, for example, a virus might bind to the protein. Okay, so I'm where am I in time? Okay, so I'm pretty close to the end. All I want to do now is to show you 2 very brief results, so we still have 5 or 10 minutes left for questions at the end. And the 2 brief results that I want to talk to you about are something called non-uniform refinement and 3D variability analysis. Non-uniform refinement just appeared in a journal called Nature Methods a month ago. And 3D variability analysis was just conditionally accepted for publication. I just want to give you a feeling for these two results. So earlier I mentioned that the rigid case, which is everything we've assumed so far, assumes that these particles are rigid, but they're not really rigid. And so in the first case with non-uniform refinement, we're dealing with the fact that the particles themselves are spatially variable. So some of it may be due to motion, and some of it may be due to regions of disorder that just are not original or rigid from particle to particle. And some of them are dealing with fractional occupancy, parts of the protein that are there in some images and not there in all images. And then the next thing I'll show you is just some brief results we have on variability analysis, where we're actually estimating not only the three-dimensional structure of the particle, but also its dynamics. Okay. So the key to non-uniform refinement is the following, that when you look at a particle, this is again another ion channel, what happens is that there's certain regions that are not rigid. And in this channel, there's a region, this blue stuff up at the top is all sorts of garbage. It's all sorts of lipids that are stuck to the protein and are not consistent at all from image to image. And they are imaged at very low resolution. You could see the resolution up here in this blue stuff is sort of like between 8 and 9 angstroms. So it's terrible resolution. And down here at the bottom, this particular protein has a tail, and the tail moves around. So in every image, it's in a different location, and it just can't be resolved. And one of the problems is that in the denoising stage of this, so remember the iterative refinement had 3 stages. You find the rotation and translation, you do a noisy reconstruction, and then you denoise it. And the way all the techniques in the literature worked is that for denoising, they used a Wiener filter. And a Wiener filter is a shift invariant filter. So it denoises structure sort of in a spatially invariant way. But what we have right here is the fact that some regions are resolved very poorly, and other regions here in the middle are actually resolved quite well, and other regions are again very poor at the top. So what we've developed is a new method of regularization that again, it uses a form of cross-validation. So we define a new parameterized family of regularizers, which are not shift invariant filters. They're more complex. They could be a neural network, or they could even just be a spatially adaptive linear filter. And these new regularizers have parameters, and what we're going to do is maximize those parameters using a cross-validation loss. So effectively, we break our data set into 2 pieces. We do a reconstruction of we do 2 reconstructions, V1 and V2. And then what we're doing is we're looking to filter V1 so that it's as similar as possible to V2. And at the same time, we filter V2 and check to see how similar or consistent it is with V1. And by minimizing this cross-validation loss, we solve for the parameters of our regularizing filter. And what this does, so if this is just a 2-dimensional slice from a raw reconstruction, this is what you get with a Wiener filter. And this is on the right what we get with our new non-uniform regularizer. In this case, this is nothing more than a spatially adaptive linear filter. But that's enough to give us dramatically different results. So this is a PFCRT, which is a chloroquine receptor resistance transporter. It's a protein that also exists in this proteasome, this parasite that causes malaria. And it's a protein that used to be used which actually used to allow chloroquine to be a good anti-malarial treatment. And it's thought that it's because of the mutation of this protein that chloroquine is no longer a good anti-malarial drug. And so by understanding what has changed in this protein, researchers were hoping to modify chloroquine to develop new anti-malarial. The problem is traditional algorithms for doing the reconstruction fail on this protein. So what you're seeing on the far left here is the output of the algorithm that I showed you a minute ago, which is our own algorithm circa a year ago. And what you're seeing right beside it is the reconstruction of our new algorithm, where again, red here means high quality reconstruction, blue means essentially garbage. Let me just zoom in on a piece of it. Oh, this is just a plot of effectively the signal to noise ratio as a function of frequency, which shows the new method versus the old method and shows the improvement in signal to noise at sort of a mid-range to high range of frequencies. But this is what it gives you. So if I zoom in on the protein, this is our old algorithm, and this is the new algorithm. And I can just animate it to give you a feeling for what that looks like. So this is the new technique that we just published in Nature Methods. And I think it's actually a real game changer in the field of electron cryomicroscopy. Okay, so that's one result. And again, this is the old technique, again with the molecular structure docked, and this shows you the new technique. Again, old, new. Okay, so that's one result. And this was exactly, this is a figure from the paper that was published back in February in Science, where the very first team ever reconstructed the structure of the spike protein. It was a group of researchers from NIH and the University of Texas in Austin. So this is a figure from their paper which showed the use of CryoSpark, which is the software from our group here in Toronto, that showed the ab initio, the refinement, and then the non-uniform refinement to resolve the highest frequency structure. And I'm just about out of time. Yeah, I'm just about out of time, so I'll just show you a couple of results. We've been able to extend this technique further to actually deal with conformational variability, which basically underpins the underlying dynamics of these proteins. And I'm just going to skip a few slides here talking about protein dynamics. But people in the bioliterature do sort of coarse scale protein dynamics using mass spring models. And it turns out that if you use these mass spring models, the resulting motion turns out to be these oscillating modes, where the low frequency modes correspond to essentially the lowest energy modes, which mean that these are the modes of oscillation that are easiest or most energetically favorable for the device or for the protein. And it also turns out that the covariant structure of the different states of the protein over time is just a scaled multiple of the inverse Hessian. This is Planck's constant and temperature here. And so basically what it says is, if you can learn a linear subspace model for the dynamics, you can actually find something interesting about the local sort of equilibrium dynamics, the dynamics around equilibrium base states of these proteins. And so we've developed an algorithm to do this, which replaces the V in the original equation by a linear combination, a linear subspace model of different components, which in combination explain your images. And so let me just show you an example. So this is a proteasome, and these are the basic motions of the protein down at atomic resolution, which show you the different modes of variation in the proteasome. Here's another example. This is a sodium ion channel, which is common throughout the nervous system, associated with spike production in nerve cells. And it shows you some of the motion of the structure. Let's see. Yeah, I mean, this is one more. Oh, this is actually, so this is the motion of the spike protein. And this was actually very important for the development of vaccines, because what it showed, it doesn't mean much to me, to be honest, but what it showed biologists is that there are two different states of the spike protein, what they call an open state and a closed state. And in the open state, receptor binding domains are available for connecting to the human ACE2 protein, and in the closed state, it's not available. So it was crucial to really neutralize this. You want to get neutralizing antibodies to bind into this open state. Anyway, I think that's all I've got. These are the 5 guys that contributed to the various pieces of this. Ali Punjani is a PhD student in my lab, and he is the CEO of the company that manages CryoSpark, our software. Marcus Brubaker was a postdoc in the lab and a PhD student in the lab. John Rubenstein is a professor at Sick Kids and U of T. Hao Wei Zhang was an undergraduate student in the lab. And Suhail Dawood is the sort of main software engineer at Structura, which makes CryoSpark. Okay, so that's all I've got.
Thanks very much, David.
Let me stop. So I know we don't have much time for questions, but I admit that I've prepared poorly and I had too much stuff. My bad.
I enjoyed it thoroughly, every aspect of it. So we have questions from students. They have priority. Can you go to the chat box, David?
Okay. Oh, I got.
So Cameron has a question.
Okay. Oh, first one's from you, son. Okay. When it comes to
I actually didn't type questions. It's from Cameron.
Okay, yeah.
He said when it comes to semantic.
Yeah, when it comes to semantic association of features and different objects of the scene, it seems to me that we're teaching robots to see like humans. Are there advantages to robots that don't try and incorporate human assumption? Yeah, boy, that's a great question. I think the way people have solved the semantic alignment problem is actually not necessarily the way people do. We don't really know how people solve that problem. And you know, the way I look at computer vision and machine learning is we're not necessarily trying to solve problems the way humans do. It's actually the opposite. We don't know how humans really solve these problems. And the problem with human psychophysics and human neurophysiology or human neuroscience is that we don't have very many good models to test. So the only thing you can do in psychology is test a model, or effectively to falsify a model. So the way psychology works or neuroscience is you propose hypotheses about how the human system works, and then you go in to test those hypotheses to see if you can falsify them. The problem is we don't have many hypotheses. So what computer vision and machine learning are good for is coming up with hypotheses in the first place. So rather than building our machine systems in order to look like human systems, I would actually argue that it's actually a symmetric sort of channel. It's a bidirectional channel where the great thing is by coming up with new algorithms, we're giving the psychologists new hypotheses that they can test. Okay, so, but now there's a second point to the question though. Are there good reasons for building machines that see the same way humans do? And the answer may be yes, because it allows the machines to then explain to humans how they came up with what they saw. And that relates to the way humans see things. And that has been sort of, it's thought that that is potentially very, very beneficial. Okay, next question. Thanks for the wonderful talk. For the large-scale scene reconstruction, it seems the point cloud reconstruction with feature points is sparse. Yeah, so that result that I showed you is from a long time ago. And we have new models now where we can actually do this totally densely. And people are using things called neural implicit functions, where they're effectively finding mesh-like models. So rather than point clouds, they're effectively finding coherent, opaque surfaces, and that has been incredibly useful. And so I'm just not fully up to date on my demos, but indeed, that's an excellent point. The next question, I have a question on image refinement on multiple molecules. Would it be possible to separate molecules from their movements without knowing the exact shape? Yeah, so that's what we effectively are doing when that last piece of work that I showed you on the variability analysis, we're simultaneously effectively solving for shape and motion, but we're doing it within a linear subspace, which is sort of, it's not quite what you want. Your question is getting at a much better way to do it, which is to say, what if you actually simultaneously estimate the shape and the actual transport, the motion of structure, the actual transport of atomic density? And that's something we're working on right now so that you can actually infer simultaneously shape and motion or conformational change. And we also have a new method which uses a probabilistic mixture model that allows you to have actually a combination of totally distinct or different particles in the same sample. And you can do that with what's called a mixture model, where it allows you to explain every image in terms of one or more three-dimensional shapes. And that allows you to deal with totally distinct shapes. So good questions. Okay, that's all I've got for now. Are there any other questions on the fly?
There's one more. It says, is the work done at U of T or with Google?
All of this work is at U of T. I don't do any cryo-EM work at Google. I've tried to keep that totally separate for a wide variety of reasons, but I really want this work to be free for the biology community to use so that we can make, you know, strides in our understanding of fundamental biological problems.
I think our students are also interested in knowing what they're doing at Google Brain. You can tell us a little bit.
Yeah, at Google Brain, who knows what I do at Google Brain? I manage a group of about 15 researchers. Jeff Hinton is one of the guys in the group, but there are many other researchers in the group who are absolutely stunningly good, far smarter than I am. We have a group that really, we do very basic research. We do, a lot of the research is focused on new neural network architectures and learning procedures. This includes architectures for computer vision, for natural language understanding. We have some new techniques on speech analysis, which look really, really super good, as well as new techniques for speech generation and natural language processing. Anyway, the group is very small, but it's extremely diverse. So everything from computer vision to machine learning for computer systems, to machine learning for natural language understanding, speech understanding, and all sorts of new neural architectures, which may or may not be, you know, the state of the art 5 years from now. It's a very fundamental research group.
So if our students want to find internship opportunities, you know who to talk to.
Yeah, so Google, they could write to me directly. In fact, what I would recommend is writing to any of the people in the Toronto Google group. We're called the Brain Research Group in Toronto. I don't know why they call it the Brain Group, but there's a larger brain. There are brain teams in several cities. There's a brain team in Berlin, and there's one in Mountain View. There's one in Montreal. There's one in Toronto. One of the best ways to get internships would be to write to the individual researchers directly.
Good. So our students, any more questions for David? I told you he's very hard to get. Don't let him go easily. So I have a very quick question, just to have the somehow, since I'm the coordinator, I have the priority. So I'm used to seeing structural structures from cryo-EM, published in Science and Nature. I'm not aware how people accurately deduce the molecular structure's movement. And you showed beautiful motions of the open state and closed state. And but I know for EM, I do a lot of EM, not cryo, but similar. It's really a still shot for a lot of particles.
Absolutely.
So are you trying to deduce from different poses and to figure out the movements?
Yeah, so that's right. So the idea here is imagine that you have this structure and the structure moves like this. So you can think of maybe it has like, let's say you have an articulated body, my arm. Then you can parameterize the articulation of the elbow maybe with 1 degree of freedom. So then you can model all the different shapes that my arm might take as a curve, right? A single curve in the operating space of my arm. Now, if it was a ball joint, if it had 3 dimensions of degrees of freedom, then I would have a 3-dimensional manifold of different states of the arm. Effectively, what we're doing when we estimate motion, we're not estimating motion per se. What we're doing is trying to find a manifold in a high-dimensional space where all the images that we have are consistent with points on that manifold. And then when I show you motion, all I'm really doing is running over that manifold where there are points. I'm sort of linearly interpolating between points on a manifold. So I'm not really estimating velocity or acceleration per se. I'm just assuming that nearby points on the manifold are connected through some kind of smooth motion, and that's what I'm actually showing you.
I see. A couple of days ago, I read a paper on the molecular structure of COVID-19 structure, and I remembered the mechanical engineering sense. The title of the figure says it has a flexible hinge, and it shows a little mechanical spring, a hinge-like structure, and the structure can move back and forth. And that somehow ties closer between biochemistry and mechanical engineering. I was very happy to see that.
No, so what got me enthusiastic about this field in the first place is that these proteins are all little nano machines. They're all little nano machines. They're highly constrained by the physics and the chemistry. And they literally do all the things machines do. They articulate, they have ratchets, right? They literally move ions through the protein going from outside the cell to the inside of the cell. I mean, they really look mechanical in nature when you get down to the atomic level. And it's really quite exciting from that point of view, because all of the sort of tools and understanding that you can think of in terms of these little nano machines, they're all there. And so understanding how they work becomes a sort of atomic scale mechanical engineering problem.
I'm happy to see David is not a pure computer scientist. He's more physical now.
Well, yeah.
But thanks very much for your time. I know I also personally enjoyed your lecture very much. I thank you on behalf of our students as well for your time. And anyhow, so thanks for your spending so much time with us.
Oh, totally my pleasure, son. Anytime for you, anything, you name it. The Institute's going well. This course, I think it'll be exciting. And I'm happy to help with any time that I have to spare.
Well, thanks very much. You'll make me look much better in front of our staff and students.
Okay, take care, son.
Yeah, I'll see you, David. Thank you.
Bye.
Thank you.